First of all, mitosis coloring. If I haven't graded that, I'll come around at some point today and check that off. We are going to practice your mitotic index skills on actual microscopes. So you will get a slide that shows you an onion root tip, and then it will have all the different stages of mitosis. And then from that, you will calculate the mitotic index. A reminder, you do have a quizzes due. It came to my attention this morning that I failed to post the link. So if you tried to take the quiz, it wasn't there, now it is, okay? But again, it's not due till Friday. You do have unlimited attempts on this one, so I'm expecting everybody to get 100%. And then you are looking forward to your test next week on both tests. All right, 2 ways mitotic index is used. Give me one. To diagnose cancer and treatment. We want to see if it's working. So if we're giving treatment, we can then do a multiple mitotic indexes, hoping that the number is decreasing, right? If the number is decreasing, that means our treatment is probably working. Very good. You guys need a colors box. I should say materials box, sorry, in the back of the room. For the microscopes, remember your microscope techniques, okay? If you always start on low magnification, get focused, and go up from there. When you're calculating your mitotic index, you can just look at what's in your field of view, okay? So you don't have to move it around and get like the whole onion root tip. Just pick a field of view, count the cells in that field of view, and calculate your mitotic index based on that. What I would like to do is get you guys on a microscope, get yourself focused, and then get a picture. And then you can use your picture to calculate. Your eyes will thank you if you are using a picture rather than looking in the microscope all the whole time, okay? So let's do that. If you're at a table with a power strip, go ahead and grab a microscope and a power cord. And if you are not, let's see, what do I want you to do in the meantime? Actually, what you could do in the meantime, I have a page on the notes. So if you can open up the notes and go to slide, however many will fit in the power strip, or however many will fit safely at the table. If you go to slide 53, there's a couple links that will help you review mitosis. So if you're not on a microscope, you're on the links from 53. Or if you haven't done your syllabus statement yet, you can work on that. You can check Aries, like use it as some work time to be productive in some way. Oh, yes.
I'm gonna get my Chromebook.
Any student-led conferences or progress reports? All right. I'm going to pause our recording until we get to our notes. Remember that interface is in between cell division. Okay, when you were counting all of your mitotic index things, you probably had a lot of interface, right? Most of them were in interface. And that is when the cell is not dividing, in between, right? Mitosis is only PMAT, and then after PMAT, kind of simultaneously a little bit, we have cytokinesis, which is the division of the cytoplasm, which is the division of the cytosol and all of the organelles except the nucleus, okay? So make sure you have those 3 sections separate in your mind. They're separate processes.
Cell division.
That's a great video. You could probably watch that on your own. We are going to draw in our notes. There is not a spot for interface. Darn it. Oh. Actually, there's a spot on page 4. Yes. On page 4, can you please draw both G1 and the S and G2? Okay, so you're doing 2 circles. And I do want this in color. Color matters. So get a blue. Colors you will need today is blue, red, green, yellow. Blue, red, green, yellow. I think that's all. Blue, red, green, yellow. Yes. On the bottom of page 4, you're going to draw G1 and then the S and G2. Blue, red, green, yellow. Oh my gosh, that reminds me of kindergarten when I was learning my colors. Is that Naranja? Oh. I'm pretty sure that's my kindergarten. And I don't think my kindergarten teacher actually orange. Okay, so what are we drawing here? G1. So G1, your outer circle is your what? Cell membrane. The inner circle is our nuclear membrane. Yes. The little solid circle inside of the nucleus is the nucleolus. Very good. And our red squiggly stuff is chromatin. Thank you for using the correct version of DNA.
Chromatin. Oh my.
So for this one, we don't have a spot to describe, so I just want you to label it, okay? So in your red color, put chromatin, and then maybe in parentheses DNA, just in case you don't know that chromatin is DNA. And then we want to label our nucleolus and our nuclear membrane. I'm going to let you skip the cell membrane because I think that's obvious. And also in mitosis, we're not really focusing on the cell membrane, right? We're focusing on the nucleus and the stuff inside the nucleus. All right, so that's what your cell looks like in G1 phase. It's okay. You can have 4 squiggles or 3 squiggles. Doesn't matter. However, the next picture does matter. How many squiggles you have? Because what happens in S phase? Not yet. S, what did S stand for? Synthesis. What are we synthesizing more of? DNA. Notice the number of red squiggles has doubled. So however many red squiggles chromatin you had in your first picture, you need to double that. They are not chromosome form yet. They're still in chromatin, but there's just twice the amount. Okay, so still drawing your nucleus, your nucleolus, the same. And if I had 3 squiggles, I'm now going to have 6 squiggles. Okay, for this one, just note to yourself that the DNA doubled during S phase. What? Oh, my bad. I'm sorry. That was stupid of me.
Oh, I was right.
You were right, and thank you for mentioning that.
I need a super hole.
Sorry. My bad.
You're Creon. Why do you think women aren't as smart as men?
This is what happens when you get less and less sleep every day. Yeah.
I lie when I get nice thinking of how Alan's like Creon.
Oh. a lot like right after dinner, and then he'd go to sleep when I went to sleep. That's no longer a thing. He just is up all night now. He sleeps a lot during the day. Yesterday, during fourth period, he had hiccups all period. He sleeps very good. No, but he moves a lot. He likes to kick me. Great. So I'm kicking from the inside, kicking from the outside. Well, he doesn't know he's kicking me. I need him back. Push him back. He falls asleep and then he goes starfish, and then he contracts back in and then starfish again. Anyways. Okay, so that's interface. Prophase. Now we are going to page 5. You are going to draw and describe. Your description is in the text here. Yes, I need you to match exactly the drawing. Oh, I would say maybe write it all in black pencil and then underline in the colors. That would probably be smart. Yes. Yeah, so I think that's a really good plan. You can write it all in pencil, but then the blue underline in blue, because these are really important things that happen. In prophase, you have to know these 4 things happen. Okay? So when you draw your nuclear membrane, we're drawing it as a dashed line now because the nuclear membrane starts to go away. It like dissolves, kind of. Same thing with the nucleolus. You're going to draw your nucleolus dotted because the nucleolus also starts to disappear, go away. Then the DNA supercoils into what we now call chromosomes. So when you were looking at your microscope slides, you saw most of them were just your nucleus was just a dense purple ball, right? But if you saw anything in the stage of mitosis, in any of the stages, you notice they look like little noodles, right? And those little noodles are visible because they were supercoiled into these chromosome structures. So in any of your pictures, you saw the little noodles, you were actually looking at chromosomes. And in all the other pictures where you just saw purple blob, you were looking at chromatin. All right. The 4th thing that happens in prophase is your spindle fibers. Your spindle fibers are in green. They're coming out of your centrioles. Okay. Technically, I probably should have made your centrioles a different color as well, but it's okay. You colored your centrioles in your mitosis coloring though. They were orange in your mitosis coloring. So in prophase, those spindle fibers start to form. Okay, those spindle fibers only come about when the cell starts to divide. All right, so let's check. I was going to say let's check your drawing, but some of you wrote first and some of you drew first, which is fine. But I'll give you another minute. Prophase has probably the most to write. Prophase and telophase. The centrioles probably would make more sense to be green, maybe. Maybe not, because you want to know that the spindle fibers are a different structure than the centrioles. So if I was going to redraw this, I would probably draw them maybe black or orange, maybe. But don't worry, it's fine. Okay, so when you redraw this 5 times for homework, you want to make sure that you draw, I would do the colors, okay? And make sure you draw your nucleus dashed, your nucleolus dotted, your chromosomes in chromosome form, okay? In the X pattern. And your spindles, you just have little baby spindles, okay? Little baby spindles. All right. For your homework, you're just drawing it, but you should be able to look at the picture and go, what are the 4 things happening in this picture? Yes. All right, are we moving on? You have 10 seconds. And if you haven't underlined the blue stuff in blue and the red stuff in red, I would suggest it. Or writing it in that color.
You know the movie Cube?
I've seen it a long time ago.
Do you think Mr. Lazier could figure out the equations going on in there?
Probably. I know that when he was growing up, that was like one of his favorite movies.
Cube. It's a pretty good movie. It's free on YouTube. Cube, but
Cute. It is good. And then I know he like had me watch it, and pretty sure he tested me to see if I could figure it out. And I was like, I don't want to figure it out. I just want to enjoy the movie. He's like, figure it out.
I saw a video of some people like trying to the whole Cube.
Really?
And he couldn't even do it because it was too complicated and it didn't make sense.
He does. He can't do it very fast though. Okay, here we go. This is where you're introducing your yellow. So let's actually all draw first. I think it's actually best to draw first. Let's draw first. So you're still going to have your cell membrane, but notice that your nucleus is gone. Your nucleus is all gone. You want to draw 3 chromosomes right in the center there. And your spindle fibers. Some of your spindle fibers will attach to the center of your chromosome. That center you colored in your coloring was the centromere. At the centromere, you have something called the kinetochore. Okay? The centromere is like a location. The kinetochore is kind of like a mechanism. So some of your spindle fibers, notice, will attach to that kinetochore, which you're kind of just coloring yellow. Just kind of highlight a little yellow section on the opposite sides of the center. Let me turn off the light. And notice that some of your spindle fibers don't actually attach because they have a good purpose as well. Not only are the spindle fibers going to attach to the chromosomes and help the chromosomes move, but they also help to elongate the cell. Okay, the cell is going to be stretching out, and the spindle fibers are doing that job too. That better? Color's better. So the yellow is just, if you're kind of highlighting the green spindle fiber just right at the middle of those chromosomes. That's how we remember this date. Also make sure that your centrioles, that you're drawing your centrioles at the edge, the 2 edges of the cell. Your centrioles stay at the edges, and then the spindle fibers just grow in. All right. And when you're writing this, you can kind of make it shorter. You could just say chromosomes line up at the equator. It's also called the metaphase plate. Down here? I would just do the picture, I mean, this drawing down here is actually a lot better, but the picture will suffice. Then your spindle fibers connect to the kinetochore at the centromere. Probably good to put the mechanism of movement. So yeah, I guess you can't really shorten it too much. Kinetochore. What does kinetic mean?
Movement.
Movement. You remember back to 8th grade, we talked about kinetic energy, right? Energy coming from movement. So when we think of the word kinetochore, we're thinking like movement core, right? And the kinetochore, like we said, that's the mechanism of movement. So I know you guys know mitosis, you've done your coloring. What's the next step? What's going to happen to these chromosomes? They're going to move, they're going to pull apart, right? So the spindle fibers and the kinetochore is what does that movement. All right, 10 seconds, 2 minutes? Yeah. No. Let's just stay here all day. By the way, I will be here at lunch if anybody needs to retake a test, turn in an assignment. I might be feeding thunder. That's something interesting to do.
Did you see the lightning yesterday?
I didn't. I was
It lit up the whole sky.
During
Inside.
football practice, there
I was
was
coming
thunder.
back
Really?
home and there
There
was
was a
flashing the lights and
Yeah.
a
It was lightning.
bunch of lightning.
I saw a bunch of pictures that people posted on Facebook.
What is lightning even?
We're gonna do anaphase real quick. As you're doing anaphase, I'm gonna bring around your drawing. On your drawing, the first box is I, interface. You can choose to draw either G1 or SG2. One of the two is fine. Okay? You can draw, or you could draw them both in the box. Up to you. So tonight for homework, I want you to draw IPNA. Okay, do those once. And then tomorrow we'll do telophase and cytokinesis, and you can practice those tomorrow night. So what you're seeing in anaphase, notice that your identical chromatids have been separated. So one of the things you'll learn is that a chromosome is made up of 2 chromatids. So those chromatids separate from each other. Notice the spindle fibers are still attached. And some of your spindle fibers still go all the way to the middle of the cell, because remember, elongating that cell. If you drew your cell a little more oblong, that was very good. I think most of you guys have done that. Good. Because not only are our chromosomes moving, but our cell is elongating. So this drawing format, I'm going to be looking for on your notes. When I grade your notes, if you don't have the drawing format complete, then you're going to lose points on your notes. I'm not going to require color, but I highly suggest. All right. Don't forget to be working on your syllabus statements. Whenever you feel ready, you can take that quizzes, which is due Friday. Anytime Friday. Actually, I think it might shut off at 11:30 p.m. But you should be in bed by then.
Friday, actually, yeah.
If it locks out, just let me know. But I think it'll still be open. All right.